Okay. Our next speaker is Lydia Wong, and she'll be talking about effects of droughts, droughts. Several by me. Thank you very much.
Hi, everyone. My name is Lydia. I'm from the University of Ottawa, and today I thought I'd talk to you a bit about some of the work I've been doing with my supervisor, Jessica Forrest, as well as a collaborator from Florida State, Brian Inouye, where we've been working with these subalpine bees and trying to understand the possible consequences of climate change for their populations. And I'll start by saying that I think we actually know quite a bit about the general relationships between climate and bees. There are a growing number of studies that have examined associations between climate variables and some metric of bee population success, mostly in the context of climate change. And of course, there's tons of variation in terms of the responses of bees to these changes, but overall, it seems that most studies are suggesting that the warmer and drier conditions that we're seeing in some areas are linked to declines in abundance, diversity, and range size in bees. And so we seem to have a pretty good idea of the realized impacts of climate change for bees at the population and community level, in that we kind of already know how bees have responded already to changes in climate. Now, what we know comparatively less about are the processes that are driving these declines, and that we've documented changes in climate and we've documented changes in bees at the level of populations and communities. But all the stuff going on between those things isn't super clear. And I think one thing that could help with this missing piece is to remember that populations and communities are ultimately made up of individuals, and that somehow it must be the effects of local climate variables on the fitness of many individuals that are scaling up to produce the patterns that we're seeing at the level of populations and communities. And yet, apart from manipulative lab experiments, we actually know very little about in these individual level processes, maybe because it means that we actually have to get out there. We have to get to know some individuals, quantify their individual fitness, and then characterize environmental variables that are directly relevant to them. And so what I'm hoping to share with you today is based on 9 years worth of my supervisor, Dr. Jessica Forrest, and many of her students getting to know individual bees and trying to understand how local environmental variables might influence their individual fitness. And we're interested in the overall relationship here, but also in the possible indirect relationships. And specifically, we know that most bee species need floral resources to survive and reproduce. And so we might expect there to be these indirect relationships where any effects of climate on floral resources would then affect individual fitness. So our study takes place in subalpine meadows in the Colorado Rockies. And Colorado has seen some exceptionally hot and dry conditions in the last few decades. And yet at the same time, even if it's been much hotter than it has been historically, these subalpine environments that we're working in are highly seasonal environments. So the growing seasons are very short. And that means that bees actually have very little time to find nests and reproduce in a given growing season. And so the possibility for warmer and drier conditions to relax thermal constraints and even benefit organisms in this region is very conceivable. And so it's not super obvious as to what these changes in climate will mean for bees in this region. So we work primarily with bees in the leaf-cutting bee family, the Megachilidae, and it's these 5 species in the genus Osmia who will be the main characters of our study. And so these species are solitary, so each female is working all by herself. And they nest in pre-existing cavities in dead wood, often in cavities left behind by other insects. They're also happy to nest in these wooden nesting structures we call trap nests that we can set up for them. And so in 2013 and 2014, my supervisor, Jessica Forrest, set up 7 sites in and around the Rocky Mountain Biological Laboratory. And at each site, she put up a dozen of these trap nests by strapping them to trees, along with a temperature logger that was taking temperature every hour. each hole in the block is lined with a paper straw, and this allows us to temporarily extract the straws, and then by holding them up to the light, we can count the number of brood cells that each female has produced, because each of these little segments is actually a ball of pollen and nectar that she's collected, an egg that she lays on top of it, and then divided by a wall that she makes out of mud or chewed leaves. And then because females would often use multiple cavities in their lifetime, we also caught females that were nesting and then marked them with paint so that we would know which nest belonged to which bee. And so from this, we essentially have one metric of individual fitness. We have lifetime reproductive output. Now, in addition to tracking nesting activity, we also conducted floral surveys at each site over the course of the summer, and we focused specifically on the plant taxa that we knew our focal species were using to provision their nests with, because we wanted our metrics of floral resources to be directly relevant to the species we were studying. And so thinking back to our interest in the possible effects of climate change for these bees and the pieces of information we might need to answer questions about that. But we have a measure of individual fitness, which is total brood cell count. We have floral resources from our flower surveys. We have temperature data from our loggers. And then for precipitation, we didn't have in situ measures of precipitation, so I used spatially interpolated data. We have all of this for 7 sites over 9 years. So over those 9 years, we really did get to know a lot of bees, 723 of them in total. And these are some of their faces, as well as some of their rear ends. Okay, so what did we learn from them? Again, there's kind of 2 pathways of interest here. There's this pathway, which is meant to capture the more direct effects between local climate variables and individual fitness. And then there's this pathway, which is meant to capture the indirect relationship mediated by floral resources. And I'll mention that we analyze these data as a structural equation model. So I'll be showing you the results of each piece of this diagram separately, but they were analyzed as a network of relationships. Okay, so I'll start with this first pathway, the putatively direct effects of climate on individual fitness. And so what we're looking at here is total brood cell production. So how many of those little cells that we counted for each individual as a function of mean summer temperature. And then the same thing, but as a function of summer precipitation. And what we find for temperature is that warmer summers were associated with decreased brood cell production. So each of those little data points is one individual bee of the given species. And it's a bit tricky to tell with the scaled axis, but that decrease actually represents a decrease from an average of 10 cells per bee in the coolest summers to 5 cells per bee in the warmest summers. For precipitation, we don't really see much of a relationship, possibly a positive association, but it wasn't significant. So for this first pathway, we seem to see some negative direct effects of warmer summers, but no effect to precipitation. And I'll note that even though I've kind of been implying that this pathway reflects the direct effects of climate, kind of alluding to the possible physiological effects of heat stress, really the processes driving this pattern aren't actually clear because we're still working with observational data. There could be a bunch of other things going on that are driving this pattern that we just didn't quantify explicitly. So things like competition, parasitism, and other variables. So moving on to the indirect pathway, and I'll start with the first part of this 2-part pathway. So what I'm showing you here is maximum floral density as a function of mean summer temperature. And what we see is this significant interaction between precipitation and temperature, where essentially the opposing directions of the red and blue lines are suggesting that it's specifically the combination of hot and dry conditions that are leading to decreased floral density. And while that does seem to make sense given our general understanding of plants and what they need, the plants in our study area are thought to be relatively drought tolerant because they're these long-lived perennials that live in very dry and rocky soil. And yet the results that we're seeing seem to suggest that maybe even these plants aren't able to tolerate the drought conditions that we've been seeing. So negative effect of drought on floral resources. For this last piece, even though the relationship might seem obvious, I think it's still important to quantify it because ultimately the strength of this relationship helps us contextualize the result I just showed you between drought and floral resources. And so looking at total brood cell production as a function of floral density, and we do see a pretty positive relationship where individuals who have more flowers of their specific floral hosts seem to produce more brood cells. And so positive relationship between floral resources and individual fitness. And so given the negative effects of drought conditions on floral resources, that does suggest that we have an indirect negative effect mediated by floral resources. That, in addition to the putatively direct negative effect of warmer conditions I showed earlier. And so that to me was a very sad thing to learn, given how marvelous these little critters are and how much I think we still have to learn from them. But to put this back into the context of understanding the processes at play here, I think what this work suggests is that these population and community level declines that we're seeing as a response to warmer and drier conditions may in part be driven by declines in brood cell production at the level of individual bees, and that in part is driven by the decline in floral resources. And so looking ahead, maybe what we might wonder about, hopefully in addition to curbing climate emissions, is what is the capacity of these species to respond adaptively to these changes in climate? And so before I end, I wanted to say that long before Western science took place at these sites, there were people, the Ute people, and they were forcibly removed in the 1800s in very unjust ways, often backed by the US government. And so this is something that I think about and want to be committed to help resolve, because I know I've taken a lot from this land myself. So that I want to say Tokoyak. Thank you to all those who came before us, including the many non-human contributors. And of course, thank you to all the wonderful human contributors too, and to you for listening. And I will do my best to entertain your questions, stories, and ideas.